So what I will do here in the beginning, I will compare this creature, which is the lamprey, which belongs to the oldest now living class of vertebrates, with mammals in terms of general organization and so forth. And what we will find is that, to our large surprise, actually a very large part of the organization is already present at this level as compared to this. And although with very much fewer neurons. So this is the basic thing. So I will talk in the initial part of the talk with a comparison between the lamprey, forebrain, and so forth, between lampreys and mammals. If we now look on mammals first, the newborn is, of course, a sweet little survival machine. It can cry for help, it can breathe, it can be fed, and several other things. But it's very immature when it's born. If you then follow the child over about a year, then it will be able to not only stand, but also walk, not with perfection. And what is important, why I have this slide, is to say that this is basically a progressive maturation. It is not a learning process. And if you train identical twins, the trained one will start walking approximately at the same time. It may be that the trained one starts in the morning and the untrained one in the afternoon. And of course, other animals are born much more mature, so they can essentially stand up and run like deers and horses and so forth. So if we now look on the capability, where do these different movement patterns, synergies, originate? We have networks in the spinal cord that can be called upon to activate walking movements. Of course, we have a brainstem center that determines when these circuits should be activated. If you happen to be a fish or a lamprey, it's still the spinal cord that generates a pattern. We have, in all vertebrates, of course, the respiration that is active throughout life by distinct circuits, preformed circuits. We have the chewing circuits that are activated, turned on from the brain, but generated by circuits located here. We have also circuits controlling eye and orienting movements. So in the superior colliculus, you can activate a limited number of cells and you generate eye movements. And depending on where you stimulate, you generate eye movements in different direction and with different amplitude and orienting movements. So that's another part. We also have in the brainstem, rather recently discovered by Arbor, that you have circuits which can be activated here that gives distinct reaching movements, and other circuits that can be combined to elicit grasping movements. So you have like an infrastructure here of a large number of different motor programs, motor synergies, motor primitives, if you like. You also have another area, the periaqueductal gray here, that when you stimulate, you activate discrete number of neurons in this structure. You can elicit escape movements, you can elicit freezing movements, you can elicit vocalization, and also maternal behavior. So you have a whole set of different patterns that can be activated in the brainstem, midbrain, spinal cord. So essentially, you have this set of different motor programs and many more. And for having all these patterns, you also need, of course, to have a structure that determines when they should be called into action or when they should not. And the basic ganglia are very important for the selection of value-based decisions, etc. If you look on, if you compare, this is clearly a mammal, and if you compare this much more primitive animal, the lamprey, we have is essentially the same type of coordination for the motor patterns available: breathing, swallowing, locomotion. Of course, the lamprey does not have legs and it does not have a lot of things, but essentially you have this. So the basic organization is that you have a number of networks. For instance, for locomotion, you have the central pattern generator network in the spinal cord. It's turned on from areas in the brainstem, the mesencephalic locomotor region. So this is a circuit to execute the movement. And then you have, of course, a forebrain circuit that determine whether the movement should be called into action or not. And there, the basal ganglia are of critical importance, and of course also input from cortex. And this is essentially a conserved structure from lamprey to mammals. And what is in the forebrain, the relative roles, it's difficult to say, but Sylvia Arbor and Costa in Science some years ago expressed approximately as follows: cortex can express a wish, but the basal ganglia will determine if it will be fulfilled. I don't think that is generally true always, but I think it's an important part. And what is the important part is also that you have the many different motor programs at this level. You could say essentially that the nervous system is like an orchestra. You have the forebrain, which acts as a director. The director will determine when all the different players, when they should be called into action, and the exact timing. And the orchestra, in some sense, can be compared to all the different motor structures that exist in the midbrain, brainstem, and spinal cord. So it's an analogy, but I mean, essentially the forebrain is often not determining the details of the movement, but it calls upon different circuits in the brainstem, spinal cord, midbrain that execute the movements. So that's a very general starting point. Now, again, back to the lamprey. So the ambition here is to compare the mammals, compare mammals with the lamprey. And the lamprey diverged then 560 million years ago. And what is common here has most likely existed, had already been formed at this point. So let me just introduce a lamprey to you. So it's a very primitive vertebrate. It has an important genetic position in evolution. It's diverged, as I already said, quite a few million years ago. It's actually not a jawed vertebrate, but it has a cyclostome mouse. And it has developed all basic features of the vertebrate nervous system, although one should say that cerebellum exists, but it's not very well developed. So the first question we will ask here is regarding the forebrain. It's our most recent work. So how much of the mammalian forebrain had evolved at the beginning of vertebrate evolution? Is there a motor cortex? Is there cortical sense representation, vision, and somatosensory? Is there a thalamus? Are there the basic ganglia? Is the dopamine system, has that evolved? And habenula. So I will take you through that, and then in the later part of the talk, I will talk about the CPG and the simulation. So this image you recognized, so essentially it has been assumed, and in the literature up till this very day, that the amniotes, mammals, birds, and reptiles, they had a cortex, whereas it had been assumed that lower vertebrates do not have sensory representation, do not have a motor cortex, etc. So that was our starting point. So, so the first thing we did now, some years ago, was that we stimulated this little region here, which corresponds to what becomes cortex in mammals. And we, to our surprise, we could show that we could elicit movements, we could elicit discrete, precise eye movements, we could elicit orienting movements, we could elicit swimming movements, and we could elicit oral movements. So clearly there is a motor area, a motor representation. The next question is this organized in a similar way as in mammals? So the way to analyze that is anatomy and physiology. So we injected dye into this area, and we looked on where we have projections. And we do have projections in the optic tectum in exactly the same layer as in mammals. To pretectum, to the mesencephalic tegmentum, we had very dense innervation of the large reticulospinal fibers as a cortical reticulospinal that we have in mammals. And we have also projections to the rostrospinal cord. So these are the same projection pattern as in high vertebrates, which was totally unexpected. The next level, okay, we have the projection. Do they make synapses? Do they make the same type of synapses? So we stimulated here and we recorded in the optic tectum and the reticulospinal fibers. And what we could see is that when we stimulated repetitively, we get a clear depolarization, and that was blocked by glutamate antagonists. So it is a glutamatergic projection, and it is monosynaptic since it remains in high magnesium calcium. And if we look on the projections to the brainstem reticulospinal fibers, we see that we get an EPSP, and which is large, and it's a very efficient activation, again abolished by glutamate antagonists. So we have, and if you record extracellularly, you have a very massive activation. So essentially, the projection pattern from cortex in lamprey and in mammals is to the optic tectum. We have this projection pattern. We have also, which I did not mention, we have the precise projection to the input of the basic ganglia, the striatum, the subthalamic nucleus, and also the substantia nigra. And we do also have the projections to thalamus. So these are the identical projection pattern as you have in mammals. And if we look on the input to striatum, it has both the direct collaterals of the deep pyramidal tract fibers, and also the intratelencephalic that are present also in mammals. So this is, in a sense, a blueprint with much fewer neurons, but with the same connectivity as you have. If you then ask, is there a sensory representation? We then used another situation. We had retina open, so we can stimulate different parts of retina to see if we get a response in lamprey cortex. And what we can see is that we get, depending on where we stimulate, we activate, we have an activation in different areas. So essentially what we have is a retinotopic representation of the different areas here. So it means that we have something analogous to V1 in mammals, sort of direct projections, and these are conveyed by thalamic fibers. If we then ask ourselves, is there a somatotopic area in the lamprey cortex? We did the same thing, but instead we stimulated the input from the head or from the spinal cord. And we can see that we also get activation. And what is important is that we get activation from the head area here and from the body here. So it means that the somatotopic organization here is a retinotopic organization here, and it's a motor area. So these are, in principle, the same basic design as we have in higher vertebrates. So, I mean, if one makes a more detailed comparison between lamprey and mammals with connectivity in motor cortex, different projections, types of neurons, etc., you can see that we have, on this level, very detailed similarity. So what it does mean is that the lamprey it does not have a 6-layer neocortex, but it has a 3-layered neocortex with the same composition of inhibitory and excitatory neuron. It has a visceral somatosensory area, which these also have. In reptiles, one has shown a 3-layered dorsal cortex and the visceral area. And birds are non-laminated, and we know less about this. So the take-home message is here that with regard to the organization of cortex, actually a lot was laid out very early in vertebrate evolution. So what about the basal ganglia? You all know that the basal ganglia is this, in the human, this large bluish structure here with stratum, which represents a large area. The connectivity here is in mammals and others, so that we have an output level, the substantia nigra reticulata, and it contains neurons that are spontaneously active at rest. They are GABAergic, and they project to a whole variety of different motor centers. It has recently been shown its specific projection to no less than 42 different areas in the mouse. So the function of this is, of course, that under resting conditions, all the different motor centers are inhibited. But so how can you elicit action? Well, one contribution is then to have what's called the direct pathway from the input level stratum, and it does inhibit sort of, it's topographically organized, but it can then inhibit cells here and thereby disinhibit, remove inhibition and permit action. So that's what is called the direct pathway. Then you have another set of straighter projection neurons that project to an inhibitory structure that is also spontaneously active. So when this is activated, this is inhibited. So you remove inhibition from here and thereby facilitate. It also projection via subthalamic. So this is, and then you have the dopamine system that is acting on this and activating this part and inhibiting this part. Now, if you do the same thing, so you do the same analysis on the lamprey. Actually, all components here are also present, and we had, when we started this, which the purpose of which was to look at selection of action, I anticipated that it should be a very simple circuit. And to our surprise, we found then that we have all components here. We have, if you look on the input to striatum, it's from cortex and thalamus, we have, and dopamine, 5-HT, etc. We have the same type of projection neurons with D1 and substance P as neuropeptides, and cephalin here, spiny dendrites, same type of ion channels. And we have striatal interneurons, at least 2 of the most common, the others we don't know. We have the output pathway here with SNR, spontaneous active, same type of transmitters. So practically in all parts, down to very detailed levels with spiny dendrites, inward rectifier, ion channels, etc., we have the same organization. And this is a large series of papers over several years. So this speaks for conservation. Let me give a little bit of the evidence for it. This is the lamprey striatum. And it has then the same sort of moderate, that has dopamine, it has a 5-HT, it has a histamine as you and I have. It has then input from pallium and from thalamus. So that's key. The cells are spiny. We have the same sort of gene expression, GABA, as the choline transferase, the D1 and the D2, and also this important master molecule activated from D1 receptors that Paul Gringard showed in Mammoths and combined. So this infrastructure is there. Then the connectivity from striatum to pallidum. We could show that the neurons here, from where striatum projects to this area, they are also spontaneously active. And they are also active if you block glutamate transmission. So they are spontaneously active, as they are in mammals and man. They're in all our brains, these cells are ticking on at a frequency of 30, 40 hertz or something like that. What is then important, if these should then take part in selection of behavior, etc., you need to have specificity. And we have specificity in that some of the neurons here project to the locomotor command regions in mesencephalon, and we have the effect on the optic tectum and also STM and the diencephalic locomotor region. So we have a specificity there. So we can say then that we have the substance of that. So it's at rest, we have an output, a pallidal output to the brainstem, motor centers, and that is tonic and inhibitor. And then is the question, is this sufficient then to activate the motor center? And what we then looked at was a connectivity that it goes from the basal ganglia, pallidum, to the locomotor command, the mesencephalic locomotor command region, which in turn via the spinal cells projects to the spinal cord. And since we know that these are chronically active, what happens if we block locally with GABAC? And when we block this, we release this from inhibition. And what you can see is that you then turn on alternating activity in ventral roots. And if we have the whole animal there, we can elicit locomotor activity. The same for eye movement, the same for other motor centers. So our concept of this here, and I think also in mammals, is that we have different modules of cells, and we have both the direct and the indirect pathway represented there, as I showed you earlier, and that you have like modules that control locomotion, that control different types of eye movement, steering and feeding, etc. And whether one of these should be called into action, it depends on the input from cortex or thalamus, and the facility. And if the dopamine system is active, as with salient stimuli and so forth, that enhances the activity of D1 and makes it more likely that the movement will come out. And of course, the lamprey has a very limited behavior repertoire. And what has happened during evolution, as we interpret it, is that with an increased behavior repertoire from lamprey to fish, to amphibians, to reptiles and mammals, you have added a number of modules in order to account for the more and more complex behavior repertoire. But the surprising thing is that one has kept the same design, the same design of the different components. So the basal ganglia has, in terms of intrinsic connectivity, being conserved, but one has multiplied the number of units. So it's a conceptual scheme of basal ganglia function. Let me just make also the point that, of course, there is a dopamine system. It's here. The same, it may be boring for you now, but it's a detailed connectivity. This scheme applies to both lamprey and rodents. SNC to the different outputs, the inputs from striatum, etc., and also the output from SNC to different brainstem structures, which is often forgotten, but it's present in rodents, it's present in lamprey, etc., and plays an important role, which I cannot speak about now. And a last thing about this, if you kill the dopamine neurons with the toxin MPTP both in humans by accident and in other mammals, you kill the dopamine neurons and you produce Parkinsons. In the lamprey, you get a very rapid reduction of the locomotor activity. It's very difficult to elicit locomotor activity, but you can if you pinch them, etc. So that is, and you have also delayed reaction time, etc. Now, this MPTP is a rather dirty drug, so it could have any other effects. But if we gave apomorphine that can then replace the dopamine, we return to a normal motor behavior. So what it does mean is that also in this case, the dopamine denervation gives the same class of symptoms with hypokinesia and prolonged reaction time and a variety of other things. So from the motor part, it looks as a Parkinson-like symptoms. So this is probably also a conserved nature. So essentially, what I've told you so far is that we have this lamprey. What we have shown is that there's a three-layered cortex, as in this, which I didn't actually show you. You have the basic ganglia that exist and in great, exquisite detail has this pattern of connectivity. And we have nigra with the connectivity, etc. And the same type of receptors, the same type of connectivity. So and I haven't talked about habenula, but for those of you that know where habenula is, this is also a complex structure for control of different modulators. And essentially, this is all there. It had been sort of blueprint of this had been invented at this point, very early in vertebrate preparation, vertebrate development. And and previously one thought that it was not there. So we have made it at least 250 million years older. So the design of the vertebrate forebrain invented very early in vertebrate evolution and maintained. Now to the more at the execution part, the CPG. The reason why I started with the lamprey was now many years ago was that I like to understand how the network generating locomotion was composed. And we had worked on the mammalian preparations. We have shown that there is a detailed CPG, that there is sensory feedback, etc., that spinal animals can be made to walk. But what was the design of the CPG? That is what we wondered. So if you think about what one needs to understand in this sort of bottom-up analysis, if you are to understand the CPG, you need first to know which nerve cells they contribute. You need to identify their connectivity, how they talk to each other, the properties of the synapses, etc. And identify their cellular properties, because some cells are pacemaker-like, others are very different, etc. So it's not enough to know that cells tick on. And you need to identify the mechanism by which they are controlled from the brainstem. Realizing that if you should do this, and if you should achieve this, one needs a simpler preparation. And we then opted for the lamprey and the lamprey spinal cord at the time, which is a natural slice of 250 microns thickness and so forth, and in which you can elicit the behavior. So we had everything we needed. And I would not tell you much about that, but more about simulation. But anyhow, the network that we showed that in each segment that generate alternating activity, in each segment you have about 50 excitatory interneurons that are mutually excite each other. So when they are activated from the brainstem by reticulospinal neurons, they start bursting. And when they burst, they activate inhibitory interneurons that are glycinergic and inhibit all neurons on the contralateral side. And then is a question. It's easy to understand that they start firing when they are excited, but why do they burst? Well, that is due to membrane properties of this, of the excitatory interneurons. So when they have been firing for a while, of course, you have activation of the cells, you have NMDA receptors that are activated here, and NMDA receptors permit calcium in, and you have the voltage-dependent channels that are active when they are spiking, etc. And that will elicit enhance a calcium level, and thereby you will elicit calcium-dependent potassium channels. So, and when you activate them, you will of course gradually hyperpolarize the membrane so that they stop firing. And if you have a high level of activity, you also have an accumulation of sodium, which will activate sodium-activated potassium channels. So the burst terminating mechanism is rather much intrinsic, whereas the excitation is from here. So there's an excitatory core of spinal glutamate neurons that generate burst activity by an interaction with connectivity and membrane properties. And we have the inhibitory neurons that generate the alternation. But if you block inhibition, you can still elicit rhythmic activity. And you also have stretch receptors that can act under normal conditions, but we will not talk about that. But having a network like this, and with the description I gave you, a way to test whether what I said would really happen would be to make detailed simulations. And we have you made detailed simulation of the lamprey, also rodent-based ganglia that Jeanette and Alex and Johannes talked about yesterday. But we have previously simulated this on the lamprey with sort of simulations based on detailed biology. And this is the other colleagues here. But now the lamprey networks that are really dating back to what I did together with Anders Landsner and Örjan Eckerberg and so forth. So we simulated these excitatory neurons in great detail. So they had the same properties as their biological counterparts, and the same after hyperpolarization when they fired. So we knew that they were very close. And the synaptic interaction was a mixture of NMDA, the voltage-dependent NMDA receptors and AMPA receptors. So again, that was very similar. And we had, in the initial, we had 5 compartments, and we had included the ion channels that we had identified in a long series of papers. And so they were incorporated here. And how do we know that we had the appropriate balance? Well, it's difficult to know, but what we could show was that the net effect was that these neurons behaved almost exactly as their biological counterpart. If we then connected them so that we had a network with reticulospinal neurons activating from the brainstem, we could then generate alternating activity in the different types of neurons. And what was important was not only that we could generate alternating activity at a given frequency, but we could cover the frequency range observed biologically between about 0.2 hertz and 10 hertz. And that told us at least that we had captured important part of the pattern generating network. And we could not have done that just by doing the neurophysiology. It was very important that we could interact between modeling and experiments. And since the late 80s, we have continuously had that interaction. Now, so far it was what happens in one segment, but in reality it's 100 segments, and there is a wave of contraction passing down the body with a phase lag. And if you take the isolated spinal cord, it will still generate this pattern. So that is essentially the pattern that drives the animal forward as a wave is propagated backwards. But the network produces this, but it can also easily switch. Also, as we heard Scott talk about, with the flexibility, so it can generate backward swimming. So if you activate the caudal part or inhibit the rostral part, it can generate the reversed swimming. So we then later made large simulation of the intersegmental coordination, where we had the approximately correct number of cells, we had a network of approximately correct number of mobile neurons, 10,000 neurons at the time, so 100 neurons per segment, and then supraspinal for activating them. And we had the established intersegmental connectivity, and we had rather complex neurons at the time. So when we did that, what we also did, we had the morphology of the cells and the varied morphology. We observed that we have some neurons that are smaller and some are a little bit larger. So there is a biological variation in size and membrane properties. And we packed these neurons at approximately correct way. And one thing one can observe then is, of course, with this variability, some neurons will start firing with a low current, a synaptic current, and others will fire at a much higher rate at the same current. So there is a variability. Some neurons fire with a low threshold and others have a high threshold. What we found then was that the network actually operated better with this variability if we gave all neurons, all interneurons, the same properties. We could generate burst activity, but with a limited range and not so stable. If we introduce a biological variation, we could instead have a much more stable rhythm extending over a much larger frequency range. So the conclusion is that we had variability. It's not a mistake on nature, it's actually a design feature. And variability is useful. Okay, this just shows the connectivity, and there is an asymmetric connectivity between excitatory and inhibitory neurons with the lengths of the axons. But if we introduce this in the model and we display the activity so that the 10,000 neurons, blue is hyperpolarized, red is spiking, and yellow is depolarized but not yet spiking. And if we look at the next phase, we have these 10,000 neurons, and they are activated. And you can see that we have rhythmic activity, and we have a phase lag along the body, head and tail. So it's now. What happens if we instead take a little part here and lowers excitability, so it has lower excitability than the rest. So essentially here we decrease excitability. These can fire, but at a lower rate than the others. So the others has to be, so you have a coordination there. So essentially, can see that in this case, by just tinkering with this little bit here, we can reverse the phase coupling along in the entire network. And actually by changing the level of inhibition or excitation here, we can change the phase lag so that that we can either decrease or increase the phase lag over the entire body. So it's, I think this is another important take-home message from the simulations, that by tinkering with a little bit of a network, you can control the network. And in this case, have sort of behavior relevant forward to backward swimming. We have also included here simulations where we have included stratum and pallidum and the locomotor command region. And when this switches on, this is inhibited, as you know, and this is disinhibited, and so which corresponds to that an excitatory drive, and you can see that that produces the alternating activity. And we have done that in greater detail. So let me just go backward. So the next level simulation we have been, so we have used the same network, but then we have incorporated in the network also a simulation of the body, as you can see in this creature here. So it's artificial muscles with length tension curves, etc., that are installed in each segment, which can then generate, and we have simulated the viscoelastic properties of water. It's Örjan Eckerberg that did that initially and just been continued by Alex Koslow. So, and you can see that the swimming movements very much corresponds to what you see in the normal lamprey. So then with steering, which can be achieved by asymmetric activation of the reticulospinal neurons from the midbrain, we just I don't seem to be very good at making them swim, but so they, but what this looks, of course, very nice, and it's very useful actually to go from having the network that you can postulate will do something, to have the simulated body and also see that you can get turning responses, you can do a whole variety of things, also utilizing the details of the descending control, in this case from the midbrain. So it's actually, for the analysis, for the biological analysis, and I'm of course driven by understanding the biology, it is very useful to be able to go from the single cell to the network and ultimately the movement. And we have actually done some robots also, but I will not talk about. So this is the same type of lamprey with a basal ganglia and swim towards blue and avoids red objects. And that's, so I will not detail how that is achieved. But with this, I hope I've given you an image first of the development of the nervous system and how much was actually invented very early in vertebrate evolution. And then a little bit about the CPG and how we went about to crack the CPG, and then how important the simulation has been in this work. And as you know from Jeanette and others, we continued that now also with the rodent basal ganglia. So let me then just reiterate: we have shown that we have this enormous conservation from here to there, with the cortex, the basal ganglia, the nigra, the habenula, etc. And with that, let me say, well, there is one aspect also, and I was asked when I accepted to give this, if I could give a little bit of advice, some sort of wise advice, which I'm not at all sure I can give. But to many of you that are postdocs, or you are in the beginning to think about your career, what you would like to do. If you like to be in science and like to, I think it's important to determine what you like to do, and to focus on a problem for which you are burning. You really feel it's important. You can dream about how to solve it. So rather than to take anything that comes along, of course, in your training phase, as a graduate student or a postdoc, you can have other priorities. But when you start your own lab, I think it's very important that you think about what you like to do, and how and whether the problem is important, of course, and whether it's solvable, whether you have the means to solve it. But I mean, I think if one should be in science, one should have the ambition and the willingness and be sufficiently interested to really try to understand. And I also have, in similar situation, I've been saying that it's important to listen to advice. But remember your own focus and do not always follow the advice. It's good to listen, but keep your focus, and rather than be distracted by going here and there. And this comment has the same sort of thing. I think you should use all techniques appropriate for your problem. But when you have a new technique coming up, use it if it's useful. But don't jump on the bandwagon and do the same as everybody else with a new technique, confirming that you can use this technique also for your own problem. So these are three things. I think the importance of independence, importance of listening to the with interest to what happens around you, but still keep your focus. But of course, that doesn't mean that you shall not take all the results you get in consideration. So even if you focus on the problem, a way to solve it, you have to take in the results that come around. So, I mean, it may be that your initial hypothesis was dead wrong, and then, of course, you have to solve that. But to have a problem that you burn for. And I don't know what more to say in this context. Jeanette asked me to comment on this, and this is my little take-home message. And let me just mention those that have contributed importantly to what I talked about. Of course, the CPG work, it's some time ago, but Peter Wallén was very important. And for the simulation, Jeanette has continued to be very important, but has, of course, a big activity of her own now. For the developmental part, Britta Robertson has been a main force. And for the basic, together with Marcus for the basic ganglia, together with Shreyas for the data on the lamprey cortex, and that is actually arranged the first approximation as is. And Shran has done a lot on the dopamine system. And Andreas' work, which has been important, but I didn't talk so much about it, it's about the visuomotor control and steering. So the modeling is particularly nowadays Jeanette, Alex Koslow that you saw earlier, and Johannes Hjort, and also Joanna Frost-Nylén, Shreya Sriganarana, Ilaria Karante, and more recently Scott Thompson and Willem too. And this little fellow has been very helpful to us. And the Finns made a stamp on them, not the Swedes, unfortunately. So thank you very much for listening, and I have no more to say now. I'm open for questions if somebody is still awake.